So we're going to change the order a little bit this afternoon just because we have a remote speaker this session. So I'm hoping that Kim Nichols can hear me, but I'm going to introduce Kim Nichols, who's currently at St. Jude's. She was actually one of the original labs to discover SAP-deficient XLP1, and since that time has really dedicated her career to working with patients with diseases akin to XLP, and she's actually going to be talking to us today about some really exciting work related to JAK inhibition in the setting of HLH. And with that, Kim, I will turn it over to you.
So anyway, I wanted to thank the organizers for inviting me to speak. Rashmi, it was really a pleasure to be asked, and it's a little unusual here giving a talk because I can't see you, but hopefully you can see me and hear me. I want to acknowledge before I start that I'm not a rheumatologist. I'm a pediatric oncologist, but as Rebecca has mentioned, over the last 2 decades, really, I've been very interested in hemophagocytic lymphohistiocytosis and, related disorders of the immune system. And so today, I look forward to talking to you about our laboratory work, which is really a preclinical investigation of JAK inhibition, as a novel and potentially more rational treatment for HLH. So essentially, the hemophagocytic lymphohistiocytosis are a genetically, diverse group of inherited and non-inherited disorders of the immune system. And these are all associated with, really a dysfunctional yet hyperactive immune response. And, I've been most interested in the genetic forms of HLH, including XLP, which Rebecca Marsh mentioned, as well as some of the other forms of inherited HLH, but, macrophage activation syndrome, which is the purpose primarily of this session, is considered to be one of the non-genetic or non-inherited forms of the disease. And I think what we learn in the inherited is more broadly applicable across the various forms of HLH. So the one thing that's been known for decades, is that numerous cytokines are elevated in the serum of patients with various forms of HLH. And these cytokines include pro-inflammatory, such as IL-1, IL-2, IL-6, IL-10, IL-12, interferon gamma, as well as, anti-inflammatory cytokines. And this has actually been demonstrated relatively recently by some work out of Asia, where you can appreciate that, there's very high levels of these cytokines in the serum of patients with primary forms of HLH. And what we all know as clinicians is that, these cytokines contribute significantly to the morbidity and mortality that's associated with disease. These cytokines serve to further drive immune activation, and they also promote, profound sepsis-like syndrome associated with hypotension, vascular leak, and bone marrow suppression. Now, among these various cytokines, interferon gamma has been shown to play a central role. This was first demonstrated back in 2004 by Michael Jordan from Cincinnati, where he showed that antibody-mediated neutralization of interferon gamma could actually lessen the onset of an HLH-like illness and prolong survival in mice with perforin deficiency, which is a genetic form of primary HLH. However, it's not completely clear that interferon gamma is the driving cytokine in all forms of human HLH. And the reason that I say that is that gene expression profiling using peripheral blood mononuclear cells from patients with the genetic form of HLH does not necessarily show an interferon gamma signature. This was completed by Janos Simeji in Cincinnati a few years ago. Furthermore, in other mouse models of the disease, other cytokines seem to drive disease either in addition to or beyond interferon gamma. So in recognition of the fact that, there are suboptimal outcomes for patients with the primary form of HLH, as well as those with macrophage activation syndrome, and knowing that, cytokines play a key role in driving hyperinflammation, my lab has been working to identify new ways to target multiple pro-inflammatory cytokines as a newer and potentially more effective treatment for children and adults with HLH. So towards that end, a few years ago, we turned our attention toward the JakSTAT pathway. And just to, briefly review, many of the cytokines that are elevated in HLH, so interferon gamma, IL-6, IL-2, they bind to cytokine receptors that signal through the Jak-STAT pathway. And following binding of these cytokines, essentially the Janus kinases are recruited to the cytoplasmic tails of the cytokine receptors. They phosphorylate these receptor tails. This allows binding of the STAT family of transcription factors. They themselves are phosphorylated. Ultimately, they homo and heterodimerize, translocate to the nucleus, where they, drive patterns of gene transcription that promote immune activation and cell survival. And so we had questioned, would pharmacologic inhibition of one or more of the JAKs lessen inflammation and improve outcomes in HLH? We chose to investigate ruxolitinib, also known as Jakafi, as a potential JAK inhibitor that we could test in our mouse models. We were interested in ruxolitinib for a variety of reasons, and those are listed here. So first of all, this is a very potent and selective reversible inhibitor of JAK1 and JAK2. So these are the key JAKs that signal downstream interferon gamma. The drug has excellent bioavailability, and it's an oral formulation. It has a very short half-life, and it doesn't accumulate over time. And while this, could be considered problematic because it means that individuals need to take multiple doses, we were interested in this because we thought that this would allow for some titration of the dose. Now, the thing about it is many of the children with the inherited form of HLH, they're treated with chemotherapy, and chemotherapy can cause rather, profound myelosuppression, and it's rather long in duration. So, we were looking for something that wouldn't really suppress the immune system and suppress the blood counts in the same manner that chemotherapy typically does. There's been no reported increase in opportunistic infections in adults taking ruxolitinib. And, another thing that we were interested in was that, this targets disease in a mechanism that's distinct from cytotoxic chemotherapy. So we thought that it was possible, these agents could synergize with chemotherapy and potentially even allow for dose reductions or elimination of chemotherapy agents. And finally, ruxolitinib, it's an FDA-approved medication. It has a very well-defined toxicity profile. Accordingly, we thought that the translational potential of our preclinical work would be accelerated by using ruxolitinib because it's already been very well studied in adults with myeloproliferative disorder and to a lesser extent in children with cancer. So to go about testing whether or not ruxolitinib might be effective, we've been working in the lab using 2 different mouse models. One of those models is a model that was developed by Ed Behrens, who I believe is at your conference. This is a model of what we call the secondary form of HLH, or the non-inherited form. This is used to mimic macrophage activation syndrome, that occurs in humans that don't have any obvious underlying mutations in the known genes linked to the hereditary forms of HLH. So in this model, mice receive repeated doses of CpG DNA. This engages toll-like receptor 9 and leads to essentially activation of the innate immune system. There's a lot of interferon gamma production, and ultimately the mice develop many of the cardinal manifestations that occur in individuals with MAS, and I'll show you some of those in a minute. Additionally, if you block IL-10, you can further enhance the development of disease, and these mice become, very, very ill, with evidence of something called hemophagocytosis, which is kind of a sine qua non of, overactive immune system that you see in this kind of group of diseases. And in our second model, we use infection of perforin-deficient mice with a virus called lymphocytic choriomeningitis virus. So this is a model of the primary or inherited forms of HLH. Following infection with LCMV, these mice develop excess CD8 T cell activation, interferon gamma production, and as with the first model, they develop many of the manifestations of disease. Okay, so to go through our work with the first model, so wild-type mice, these are mice lacking any known genetic mutations, receive intraperitoneal injections of cpDNA every other day for a total of 5 doses. Beginning at or around day 4, mice are treated or not with ruxolitinib at 90 milligrams per kilograms orally twice a day, or with vehicle as a negative control. We chose this dose of ruxolitinib based on prior work demonstrating that it was effective in mouse models of myeloproliferative disorder, so JAK2-driven MPD. And ultimately, on day 10, the mice are euthanized and they're evaluated for the signs of HLH, or macrophage activation syndrome. So what you can see here is that following treatment with CPG, the mice develop enlarged spleens. Okay, so this is shown as a picture here on the left, and then quantified over on the right, where you can appreciate that there's a significant increase in the spleen to body weight ratio in mice receiving CPG injections. However, in those animals that receive CPG as well as ruxolitinib treatment, there's a complete reversal of spleen size, back to that observed in essentially normal mice that were never treated with CPG DNA. And, mind you, this occurs after only about 3 to 4 days of treatment. Mice injected with CPG also developed cytopenias. Okay, so that's demonstrated here, where there's a reduction in peripheral blood white count, in the red cell count, and hemoglobin. So these are measures of anemia, right? And the platelet count. And once again, following treatment with ruxolitinib, you can completely reverse each of these cytopenias. CPG-injected mice develop elevated levels of cytokines in the serum. So shown here as elevation of interferongamma, IL-6, IL-12. IL-10 is also increased, likely as a countermeasure to balance interferongamma. And following treatment, we see, a significant reduction in the levels of each of these cytokines. So given the positive effects that we saw on cytopenias and hypercytokinemia, we were interested in investigating whether treatment might also ameliorate some of the tissue inflammation that occurs in this model of disease. So, following injection with CPG DNA, mice essentially develop lymphocytic infiltrate in the liver and the spleen. Here I show you the liver. Sorry, I lost my mouse. And you see these clusters of lymphocytes within the parenchyma of the liver. On the bottom, we just have a computer-enhanced image where we used a specific program to identify the lymphocytic infiltrates and to help us to count them. These were essentially defined as, lymphocyte clusters containing more than 7 immune cells. We then quantified basically the number of foci across 5 low-power fields, so kind of averaged across 5 low-power fields, and we also measured the area of the fields encompassed by the foci. So following CPG treatment, you have an increase in the absolute number of foci, as well as the area of the field encompassed by those foci. And following treatment, there's a complete normalization of intrahepatic inflammation. So this was all, very promising to us, and accordingly, we then went on to test it in our model of the primary form of the disease. So in this model, perforin-deficient mice are infected with LCMV Armstrong. So this is a strain of the virus that induces an acute infection. Again, added around day 4, mice are treated with ruxolitinib or vehicle. Now, in both the CPG model and the LCMV Armstrong model, I guess I should emphasize here, we chose this point of treatment because at this point, mice have already started to demonstrate evidence of disease. We chose not to start, preemptively, because that wouldn't really be reflective of the clinical situation where patients often present with signs and symptoms of disease. We also chose, not to treat right at the very end of the course of the experiment. However, I will say that similar work to what I'm describing to you now on the perforin-deficient model was recently published by Geneviève de Saint-Basile from France, where they chose to start treatment much later in the course of the disease, and they found a very similar outcome. So these mice were evaluated between days 10 and 12 for the signs of HLH. So as with the CPG model, you can appreciate that the mice that are infected with LCMV develop a marked splenomegaly, shown here, quantified here. And following treatment, spleen sizes were reduced. They weren't reduced quite to the same extent that we saw in the CPG model, but there was a significant reduction in spleen size. These mice develop anemia, and thrombocytopenia. I will say, the white count sometimes goes up, sometimes goes down. It's a little bit less consistent in this model. However, following treatment with ruxolitinib, there was a restoration of the red cell and platelet counts, back to levels that were indistinguishable from uninfected mice. These mice also develop elevated levels of serum cytokines. It's a little bit different here. You don't see IL-12 elevated like you see in the CPG model, but you see a very marked increase in interferon gamma. So this is in a nanogram scale compared to the CPG model where we're in the picogram. So, the levels are at least 150 to 300-fold higher than what we see in the CPG model. Again, treatment with ruxolitinib significantly diminished the levels of the pro-inflammatory cytokine, at least interferon gamma and TNF-alpha. IL-6 wasn't changed in this model as it was in the CPG model. CPG model, it went back to baseline. Looking at tissue inflammation, you can appreciate again, right, there's a very significant amount of hepatic inflammation that occurs following LCMV infection. And again, this is very significantly reduced in the ruxolitinib-treated mice. Importantly for us, we wanted to see whether treatment would translate to improvements in any of the clinical manifestations. Now, I will tell you that the CpG-injected mice, they don't look particularly sick unless you block IL-10. The LCMV-infected mice, on the other hand, they untreated mice lose a significant amount of weight, up to 20% of their body weight following 1 week after infection. And here you can appreciate that the treated mice, appeared not to lose weight and were very similar to the uninfected controls. And importantly for us, really, what we found is that compared to untreated mice, all of which died by about 2.5 to 3 weeks following infection, none of the treated mice died. So these, experiments kind of collectively really encourage us to think that JAK inhibition using ruxolitinib, is a potential agent that could significantly lessen the morbidity and mortality associated with this group of disorders. And in the last few slides, I just want to show you some data on, mechanism. In the perforin deficient model, we were interested in determining whether treatment with ruxolitinib might influence the number, phenotype, or function of CD8 T cells, which are real drivers of disease in this model, somewhat different from the CPG model. So following infection with LCMV, what you can appreciate actually is there's a very large increase in kind of the number of activated CD8 T cells. And so this is kind of shown here in terms of frequency and absolute number. I'm going to move my little faces on the screen. And there's also an increase in the frequency and number of antigen-specific cells. Following treatment with ruxolitinib, there's a significant reduction in, the frequencies and absolute numbers of these cells. And finally, to look at functionality. So in these experiments, splenocytes are removed from the various cohorts of mice, so untreated, infected, and treated. They're placed into culture with HLA-restricted, LCMV peptides or MHC-restricted peptides, either class 1 or class 2. Those peptides are presented on the antigen-presenting cells to the antigen-specific T cells, and those T cells then, produce cytokines in vitro. So what you can see here is that when you take splenocytes either and put them in culture, and you look then at the CD8 T cells or the CD4 T cells, the cells that come from LCMV-infected mice produce increased amounts of interferon gamma and TNF-alpha. This is shown here and demonstrated here. Okay, so CD8, CD4. However, if you take the splenocytes from mice that had been infected and treated with ruxolitinib, I think it's very exciting to see that in vivo exposure to the drug significantly reduces cytokine production in both CD8 and CD4 T cells. We've gone on to do, quite a bit of additional studies, but I know that my time here is relatively short, so I will conclude, and I can explain more if needed later. So from the studies that I've shown you here, I feel that ruxolitinib, at least in our preclinical models, lessens the manifestations of HLH. It can ameliorate the clinical and laboratory features. We see this in complementary murine models of primary and secondary HLH. who's also shown excellent results in the CPG model where IL-10 is blocked. Our mechanistic studies show that at least in the perforin deficient model, treatment with the drug lowers T cell numbers. It lessens T cell production of cytokines, some of the key driver cytokines in HLH. Importantly, we found no effect on T cell degranulation or cytotoxicity. So my hope is that this will dampen T cell numbers and cytokine production, but preserve the ability of T cells to kill and thereby kind of allow some degree of protection against viral infections. We've demonstrated that treatment inhibits STAT1-dependent gene expression. I did not get to show you that, but this is consistent with its known mechanism of action. And again, in data that I didn't get a chance to show you here, we find that the drug in many ways operates via mechanisms that extend beyond interferon gamma neutralization. So I know that interferon gamma neutralization is a key study that's being discussed at your meeting. I think it's likely to have excellent effects in a number of patients. But, for those who may not respond to interferon gamma neutralization, JAK inhibition may provide an alternative strategy for treatment. And at least in our mice, we find that treatment with the drug enhances survival. My hope is that we can move this forward in the form of a clinical trial and test whether we see similar effects in children and adults with either the genetic form of HLH or with macrophage activation syndrome. And so, these are just the folks that helped me with my work. Ed Behrens was a great collaborator. Rupali is a former postdoc. This is my team at St. Jude. I really appreciate your attention. And Rebecca, if there are any questions, I'm happy to answer them. Otherwise, I really appreciate everyone's attention, and I hope we can turn this forward in the form of a clinical trial in the near future.
So thank you, Kim. That was fantastic. Everyone's clapping for you.